Yeah, good afternoon, everyone. I'm thrilled to introduce the speaker of the 15th ENIGMA-Sleep talk, Professor J Iliff. Together with Professor Michael Niedergaard, Jeff is a pioneer researcher in the field of Glymphatic system. I think like 12 years ago, they discovered the link between Glymphatic system and brain clearance, mainly in the during the sleep. He's also working on the aging brain, Alzheimer's disease, and also traumatic brain injury and assessing lymphatic system on those conditions. And he's currently a professor at the University of Washington. I just learned that there are two universities in Washington, Washington University and University of Washington. So yeah, then the title of his talk is Glymphatic impairment links traumatic brain injury with long-term post-traumatic outcome. We have 15 minutes to 20 minutes at the end to discuss if there's any question. Thank you again, Jeff, for accepting my invitation and joining us. So the stage is yours. Thank you so much.
Thank you. Thank you for the invitation. Make sure. All right. Can you see that?
Yes.
The unfortunate thing of the post-COVID era is every single talk now starts with those words instead of other words. Yeah, thanks for the invitation. Looking forward to sort of talking through where the glymphatic field is at now 12 years after its initial description in rodents. I'll be talking through, there's sort of two sides to this talk. The first is how do we assess the role of the glymphatic system in neurodegenerative diseases generally and Alzheimer's disease in particular, which is really the space where it's gained the most interest and notoriety, I think. And then in the second bit, I'll talk about some work that we've been doing, trying to evaluate whether glymphatic dysfunction is relevant to the chronic outcomes and chronic sequela that persist after traumatic brain injury. And I'll specifically be talking about blast-related traumatic brain injury in military veterans, just because that's something that we've been studying and publishing on a little bit recently. A couple disclosures, relevant ones are I serve as the chair for the scientific advisory board for a company called Apply Cognition, which is operating in the glymphatic space. I do receive compensation and have equity in that company. And depending on where the questions go, I may be presenting data from data generated by that company. So that's sort of up in the air. I'm a full-time US federal employee. Hope that you won't hold that against me, but that does mean that the opinions that you'll hear are mine and not that of the government, which I don't know if that makes me more or less credible. We'll find out. So when people talk about the glymphatic system, the interesting thing these days is that if I were to take 10 people and ask them about it, 10 glymphatic interested people, and ask them what it is, I would probably get 8 or 9 different answers. There's a good deal of diversity in terms of what people are talking about when they talk about glymphatic clearance. So I want to start just by level setting that and help you to know what I'm talking about when I use this term. And you can agree or disagree with that terminology, but at least you'll know what I'm talking about. So over the last 12 years or so, work initially in rodents, but increasingly in humans, has demonstrated that the fluid outside the brain in the subarachnoid space, so the cerebrospinal fluid, and the fluid inside the brain between CNS cells, so the brain interstitial fluid, exchange more rapidly and more extensively than was widely thought previously. That exchange between the CSF and the interstitial compartment is now regarded as being anatomically organized along the axis of the cerebrovasculature and rapid. Data suggests that it is driven by periodic things like cardiac pulsation and low frequency vasomotion and maybe synchronous neural activity. It appears to be supported by astroglial water transport. So astrocytes, which are the support cells of the brain, express a great deal of a water channel called aquaporin 4 that enjoys polarization to the perivascular end foot processes, so the parts of the astrocytes that face the blood vessels. that's where a lot of the aquaporin 4 is, and we think that perivascular aquaporin 4 is an important regulator or an important enabler of this process. It appears to be more rapid in the sleeping brain than in the waking brain. It appears to be coupled to meningeal lymphatic drainage. So the glymphatic system began to be described in 2012. 3 years later, in 2015, Yoni Kipnis's group, now the other Washington University, the one in St. Louis, he was at UVA at the time, described meningeal lymphatic vessels. So those are lymphatic vessels embedded within the meninges around the brain that do seem to participate in the clearance of solutes out of brain tissue and out of the CSF space. The evidence to this point suggests that perivascular exchange from the brain into the CSF and meningeal lymphatic clearance from the CSF out does seem to be coupled in some way, both anatomically and perhaps physiologically. And this process has been implicated in the clearance of amyloid and tau and synuclein, which are implicated in different neurodegenerative conditions. You shouldn't ever take any speaker's word for things, I don't think. I think you should evaluate things for themselves because I'm not showing you any data whatsoever. So if you want to see the papers that support this framework, you can go to this QR code and that'll take you to a PubMed list that shows the papers that I think support this model of perivascular glymphatic exchange. So a big question has arisen, I guess not a question, there has been a widespread assumption since 2013 that because there's evidence that the glymphatic system contributes to the clearance of things like amyloid and tau, because it seems to be sleep active, perhaps its failure contributes to the development of conditions like Alzheimer's, conditions that are themselves clinically associated, both during the period of cognitive decline, but also in the period preceding cognitive decline with sleep disruption. So beginning after the initial rodent studies, there's been a lot of jumping up and down about, oh, this is one of the basic mechanisms underlying different neurodegenerative conditions. It's a focal point or convergence point of a bunch of different risk factors leading to neurodegenerative diseases. Maybe this is the mechanism underlying the clinical association between sleep disruption and neurodegenerative diseases and Alzheimer's disease. I am a scientifically conservative person, sort of by nature and by temperament. And so my big question is that, do we really have evidence for that? Is the state of the science such that it aligns with what people are saying? Like, so if I go to the American Academy of Neurology, or if I go to the AAIC International Alzheimer's meeting, I will hear in talks, glymphatic dysfunction underlies Alzheimer's disease. And my question is that really true? Do we actually know that for sure? So what I'd like to spend the first little bit of this talk moving through is, where are we at in terms of answering that question? Do we know that Alzheimer's glymphatic dysfunction underlies the development of clinical Alzheimer's in human populations? So a couple of things that I think are important to focus on as we evaluate that question. First, neurodegenerative conditions generally are characterized by protein misaggregation. Different neurodegenerative conditions are associated sometimes with different types of misaggregates, right? So in Alzheimer's disease, we see extracellular amyloid beta plaques. We see intracellular aggregates of hyperphosphorylated tau. We see tau aggregates in other conditions as well, like chronic traumatic encephalopathy and Pick's disease and frontotemporal dementia. We also see aggregates of different proteins like alpha-synuclein in conditions like Parkinson's or Lewy body dementia. We see other types of aggregates too, like TDP-43, which appears in ALS, but also seems to be comorbid with other conditions as well. So we see this process of protein misaggregation. We also see across neurodegenerative conditions that the risk of them is influenced by not just a number of genetic factors, but a number of non-genetic risk factors. And several of these risk factors are held common across neurodegenerative conditions. So these are things that seem to confer risk to several neurodegenerative conditions and are believed to promote the development or drive the development of the pathology underlying those conditions. So these non-genetic risk factors include aging, chronic comorbidities like hypertension and diabetes, the presence of cerebrovascular disease, so small vessel disease, traumatic brain injury, and there's some data for moderate to severe TBI, and there is a level of data, though it's a little bit more equivocal, surrounding the occurrence of mild traumatic brain injury, so concussions. And chronic sleep disruption has emerged a little bit more recently as a potential midlife risk factor for the development of dementia later on in life. So in assessing the role of glymphatic dysfunction in Alzheimer's disease, we can look for evidence in animal models, and we can look for evidence in human models. I don't think that evidence in animal models can give us can establish a causal relationship in human clinical diseases, particularly in neurodegenerative conditions. Animal models have proven particularly poor in predicting what we see clinically. So therapeutic approaches that work in animal models of neurodegenerative disease, this is also true of traumatic brain injury and stroke, frequently don't translate into humans. And so we have to sort of take what we see in animal models with a level of caution. But there are things that we can learn. So that includes, do we see that glymphatic system actually contributes to the clearance of amyloid and tau in animals? So do we see evidence for that? Do we see that glymphatic dysfunction is present in animal models of these non-genetic risk factors? So in aging, or in the setting of hypertension or diabetes or cerebrovascular disease or TBI, do we see that in animal models of those factors that glymphatic function is impaired? And we can do something in an animal that we can't really do in a human. We can evaluate whether glymphatic dysfunction is sufficient to promote the development of Alzheimer's or neurodegenerative condition associated pathology. So if I experimentally impair glymphatic function, do I promote amyloid and tau pathology? So each of these, so where we're at in the field, I think, is that each of these questions has actually already been pretty well answered in published data. And so because all of that data is published, some of it for quite some time, I'm only going to sort of breeze through it at the vignette level and give you examples of how these have been answered. So the first question is, does the glymphatic system contribute to the clearance of amyloid beta or tau? This was actually something that we tackled in our very first paper describing the glymphatic system back in 2012. We observed that if you inject in exogenous amyloid beta 40 that was fluorescently tagged, it would transit through brain tissue along perivascular spaces. We also observed that if you inject in exogenous amyloid beta into brain tissue, it's cleared with a certain kinetics. So in a healthy young wild type animal, about 70% of the amyloid is cleared in the first hour. That's the blue line down here. If we knock out the astroglial water channel, aquaportin 4, which has been implicated in this perivascular transport, we see that the clearance of amyloid beta is markedly reduced. We also see the impairment of amyloid clearance in other similar types of settings where glymphatic function is impaired, such as in acute sleep deprivation and with the administration of pharmacological inhibitors of aquaportin 4. So this data and other data that I'm not showing you because you just don't have time does seem to support a role for perivascular transport in the clearance of amyloid and tau, and other more recent data suggests the same is true for synuclein. So check. And this is old data, which is why we're kind of moving quick. Second, is glymphatic function impaired in animal models of non-genetic AD risk factors? This is the place where there's probably been the most work. So there have been published studies in rodent models of aging in cerebrovascular disease and aging rodent models of traumatic brain injury, acute and chronic sleep disruption, all of which show that glymphatic function measured by intracisternal CSF tracer injection, so measuring the movement of solutes into brain tissue, by measuring the clearance of interstitial solutes out of brain tissue are impaired across these different conditions. So this is one of the first studies that we did demonstrating this. This was in the setting of aging. So this is a simple experiment where we looked at healthy young mice. So these are 2 to 3 month old mice. And then 12-month-old mice, that's a kind of a middle age for a mouse. And then old-ish mice, so these are 18-month-old mice. In these settings, we observed that the movement of CSF into and through brain tissue was slowed with aging. And there's nothing else wrong with these mice. There's no comorbid condition. These are just old mice. And then we also saw that the clearance of amyloid out of brain tissue was also slowed as the animals aged. So if we inject in radiolabeled amyloid beta, we see that in healthy young animals, maybe 70% of it's cleared in the first hour. In 12-month-old and 18-month-old animal, we see a stepwise slowing in that process. And again, this is just one setting where we saw that we and others have seen the same sort of results in many, many other settings as well. So it does appear that glymphatic function is impaired in animal models of non-genetic AD risk factors. So the last question is that we can really, I think, address well in animal models is if we experimentally impair the glymphatic system or glymphatic clearance, is it sufficient to drive the development of Alzheimer's related pathology? So either amyloid or tau pathology. Other groups and other studies have looked at synuclein as well. And we see that the answer to that seems to be yes, too. So this is one paper we published this about a year and a half ago, but this is one of probably 4 or 5 different papers that have shown the same thing with different interventions to impair glymphatic function and in different transgenic rodent models of either amyloid or tau pathology. So in this design, it's pretty straightforward. So we're using a transgenic mouse line that spontaneously forms amyloid plaques. So this has 5 different familial Alzheimer's disease causing gene mutations. So they develop amyloid plaques pretty fast. We then cross them into the aquaporin 4 null line, producing amyloid-producing mice with and without aquaporin 4. And we can measure how much amyloid is present in these brains at a relatively young age. And we see that in animals with aquaporin 4, so these are animals with intact glymphatic function, we see a certain level of plaque burden that is native to this transgenic mouse line. We see that if we delete aquaporin 4, we see more amyloid plaques. In the same study, we also evaluated not a global knockout of aquaporin 4, but actually a mouse that has aquaporin 4, but it doesn't have its perovascular localization. And we saw the same phenotype. We saw that if you lose the localization of this water channel, not the channel itself, but actually just where it's supposed to be, we see the same phenotype. And actually, the phenotype may be worse in that animal, suggesting that if you impair glymphatic function, in this case genetically, but other people have done it using some other approaches, either surgical or pharmacological, it is sufficient to promote the development of amyloid or tau pathology. So check. So that's a pretty complete I think package for what we can do in animal models of glymphatic function and glymphatic dysfunction. The trouble is, what this demonstrates is that there does seem in rodents to be a link between whether the glymphatic system is functional or dysfunctional and the development of Alzheimer's associated pathology. What this doesn't say anything about is whether glymphatic dysfunction in humans contributes to the development of Alzheimer's disease in humans, right? Our field, the translational neuroscience field, is full of situations where things are observed or observed to be effective in animal models that don't end up being the case out in human populations as well. So as we start to think about human clinical studies, we'll go back to our framework, and our framework is Alzheimer's disease is associated with the misaggregation of amyloid and tau in particular. So those are the histopathological hallmarks of Alzheimer's disease. It's influenced by different non-genetic risk factors, which we've already touched on before. And so there's a series of, I think, 5 questions that need to be asked and answered for the role of glymphatic dysfunction in Alzheimer's disease, the causal role to be established, right? And these are sort of levels of proof. First is, do we see, when we look into the human brain when we look in the brain of people who died with Alzheimer's disease, do we see the same kinds of histological changes, particularly in the at the glial vascular interface, in humans with Alzheimer's disease that we saw in mice? So is there evidence for some sort of corollary relationship between what we see in animal models and what we see in the human condition? This doesn't demonstrate a role in Alzheimer's disease, but it demonstrates a link between what we saw in rodents and what we see in humans. Second question is there actual evidence that glymphatic function contributes to the clearance of amyloid and tau from the human brain, not just from the rodent brain? This is I think this is a pretty important question to answer. If glymphatic dysfunction contributes to the development of Alzheimer's disease, then you would expect that naturally occurring genetic variation in the genes who encode the proteins that define glymphatic system, they should confer either risk to or protection from the development of either Alzheimer's disease itself, dementia, or different endophenotypes of Alzheimer's disease, right? So it should influence the neuropathological burden. It should influence rates of cognitive decline. It should influence rates of cortical or hippocampal atrophy, for example. So do we see genetic linkage between the elements of the glymphatic system and Alzheimer's disease in clinical population? Do we see that people who have these risk factors, do we see evidence of glymphatic impairment in these kinds of populations? So if we look in people with obstructive sleep apnea, do we see impaired glymphatic function? If we look in people who are old or who have cerebrovascular disease, do we see evidence of glymphatic dysfunction? And last, this is sort of the smoking gun question that establishes a true causal relationship in human populations. If we modulate glymphatic function in people, do we modulate amyloid and tau clearance and pathology? And this is the $100 million question, right? And until this question is answered, all of our data are corollary and not causal data. But this is, of course, the very hardest question to answer, right? And so this will probably be the last question that gets answered. So let me tell you where we're at. I'll show you some data from a published study. I'll refer you to some other data, and then we'll stop talking about Alzheimer's disease for a little while. So first, do we see the same sorts of changes in the human brain in the setting of Alzheimer's disease that we see in rodents, right? So do we see some sort of correlation between what we see in a mouse and what we see in humans? So let me tell you a little bit about aquaporin 4. So aquaporin 4 is this astroglial water channel. I talk about it a lot, just mostly because we identified its role in glymphatic function pretty early, and frankly, we've had a lot of grant money to support evaluating aquaporin 4. So we have a lot of data surrounding this particular molecule. Aquaporin 4, it's an astroglial gene, meaning it's expressed almost exclusively in astrocytes in the brain, not completely exclusively, but almost exclusively. And it enjoys a localization in astrocytes that is perivascular. So this is the level of a single astrocyte labeled with GFAP, which is kind of a cytoskeletal element. And you see almost all the aquaporin 4 in a healthy young brain is perivascular. It's the part of the astrocyte that surrounds the cerebral vasculature at the level of the microcirculation all the way out to the large vessels. What we observe in older animals, and these are older animals that exhibit impaired glymphatic function, as I showed you before, we see that aquaporin 4, we don't lose its expression, but rather we lose the exclusivity of its perivascular localization. So it goes from being largely perivascular to being kind of all over the fine processes of the astrocytes. And we think that what's happening here is that these astrocytes are losing their ability to faithfully route water into and out of these perivascular compartments. So if I think about water transport through this individual cell, it's going to be biased toward this perivascular space, whereas water transport through this cell is going to move every which way across the fine processes where aquaporin 4 is, and also across the perivascular infant. So in the aging brain, we think that the astrocytes are losing their ability to specifically route water and solutes into and out of these perivascular compartments. So we can evaluate aquaporin 4 localization in brain tissue. This is postmortem frontal cortical tissue from individuals that passed away cognitively normal. So these are people who are cognitively intact. These are all people over the age of 65. People who passed away with clinical diagnosis of mild cognitive impairment, and people who died with histopathologically confirmed Alzheimer's disease. When we look into the frontal cortex, we do see perivascular localization. We do see that in the setting of Alzheimer's disease, we see this non-perivascular enhancement. So parts of the astrocytes that are not facing the blood vessels, we see a bunch more aquaporin 4, just similar to what we see in rodents. And we can calculate the ratio between the aquaporin 4 that is perivascular and the aquaporin 4 that is non-perivascular. And we can form a ratio out of it. And what we observe is that the perivascular localization of aquaporin 4 does seem to decline in the setting of Alzheimer's disease. Is this related to not just diagnostic status, but also function? The answer seems to be yes. So if we look at the relationship between perivascular aquaporin 4 and localization and Alzheimer's pathology, so here the CIRAD score, this is a standardized score for evaluating neuritic plaques. So these are amyloid plaques with tau neurites in them. We see that declining perivascular localization of aquaporin 4 is associated with increasing cortical neuritic plaque burden. If we measure phospho-tau levels in the frontal cortex, we also see that declining perivascular localization is associated with higher levels of pathological phosphorylated tau. So lower perivascular localization of aquaporin 4 is associated with Alzheimer's pathology. And then when we look in people, these are people not with end-stage Alzheimer's disease. These are people who are either cognitively intact or have mild cognitive impairment. So if they have Alzheimer's pathology, they're at the earlier stage of the neurodegenerative process. We do see an association between declining perivascular localization and declining cognitive function, measured with the mini mental state examination or the clinical dementia rating sum of boxes scale. So for the top one, a lower score is worse performance, and for the bottom one, a higher score is worse performance. And so we do see an association between lower perivascular localization and worse cognitive and functional outcomes. So these data do suggest that we see the same sorts of cellular changes in the setting of human Alzheimer's disease that we see in rodents, and that those changes are themselves related to both local pathological burden and cognitive and functional decline in these clinical populations. But of course, these are corollary data, right? They don't show us causal relationships. So like we can't dissociate the possibility that pathological burden is changing astroglial phenotypes in aquaporin 4, or that aquaporin 4 is changing pathological burden, right? So we don't know which way those arrows go. That's not what a histological study can show you. Where the sort of next steps go is, does glymphatic function contribute to the clearance of amyloid and tau from the human brain? Are there genetic associations between gene variants related to glymphatic function Alzheimer's disease risk? I will tell you that while the asking of these questions has really just begun in the field, there are a few studies out there that have started to show affirmative results. So does glymphatic function contribute to the clearance of amyloid and tau from the human brain? There are 3 published studies now that provide evidence for this, or 2 published studies and 1 preprint. And the preprint just went up on medRxiv like a couple of days ago. So these are 2 published articles if you're interested in them. One is from a group at Washington University. One is from a group in Oslo. They demonstrate that sleep active clearance does seem to contribute to the movement of amyloid and tau into plasma. And this preprint suggests that, this is actually from our group, suggests that glymphatic function specifically is responsible for the movement of amyloid and tau from the human brain out into plasma in a human population. This is a long story and it's a technical one. I would be glad to talk about this some other time, but I just don't have time to talk about this and traumatic brain injury. So if you want to check this out, you can go check it out at this QR code. It'll take you to the medRxiv link. There are also a few studies, I think about 4, that provide evidence for variants in the human aquaporin 4 gene and different Alzheimer's disease endophenotypes. So there's one study that suggests the link between different single nucleotide polymorphisms in aquaporin 4 and cognitive and structural changes in the ADNI1 or in the Alzheimer's disease neuroimaging initiative cohort. There's an interesting study from the ABLE study out of Australia suggesting that aquaporin 4 variants moderate the relationship between poor sleep and amyloid burden measured by PET. Our group published an earlier study showing different aquaporin 4 SNPs are associated with longitudinal cognitive impairment in Alzheimer's disease populations. So there does seem to be emerging data suggesting a linkage between gene variants in the elements of the glymphatic system and Alzheimer's disease features in human populations. So you can go check that out if you want. This is a different QR code leading to some of those studies. This question of whether glymphatic function is impaired in Alzheimer's disease or in the setting of Alzheimer's disease risk factor gets at the question of how you measure glymphatic function in human populations. And this is a real friction point in the field right now. So the most common approach for measuring glymphatic function in humans is with MRI. But there's probably 10 different methods being used worldwide for measuring what people say is glymphatic function. The gold standard is probably contrast-enhanced MRI after intrathecal gadolinium injection. And the group in Norway and Oslo is the group that is doing the most of this. But then many other groups are using techniques like diffusion tensor imaging along a particular perivascular compartment, or other diffusion-based approaches, or ASL-based perfusion approaches, or structural MRI approaches. So there's all these different approaches people have taken to try to capture glymphatic biology. And there is emerging data to suggest that using these MRI measures, you do see evidence of impairment of these processes in the setting of Alzheimer's disease risk factors. But the big question is, which of those MRI techniques are actually measuring glymphatic function and which are just measuring other stuff? And we don't actually have an answer to that. So this is why this gets an orange check mark. I think that there's a lot of data in the field. It's just unclear which of that data is good and which isn't very useful. So the field is sort of at a crossroads of trying to understand that and try to get a sense of where that goes. And of course, the smoking gun question, which is, does modulation of glymphatic function improve amyloid and tau clearance and pathology in humans? This has not yet been answered. And the people that can answer this question, they win. They're in so many different ways. If this turns out to be true, this will be the study that will establish a role for glymphatic impairment in Alzheimer's disease. It is likely that whatever approach they take to modulating glymphatic function could be commercially very valuable, right? Because where does all this sit in the pathogenesis of Alzheimer's disease? So this is what's termed the Jack curve, which in the United States we are required by law to put into all of our Alzheimer's disease talks. This is a slide that shows what is believed to be the stereotyped progression of the pathological and clinical features of Alzheimer's disease. You'll note in black here at the far right end of this curve, this is clinical cognitive impairment and dementia, right? So this is the thing, when you think of Alzheimer's disease, this is what you think of. You know, this is your aunt or your uncle, your grandparents, or your parents having cognitive impairment that lands them in the memory clinic and eventually will end up requiring their move to memory care, or wherever it is that wherever they experience their decline. The trick is the neuropathological processes underlying this cognitive impairment are believed to begin perhaps decades before the onset of these clinical symptoms. So amyloid plaques, evidence of them measured by either PET or by CSF biomarkers, appear to emerge 15, 20, or maybe even longer periods of time before the onset of clinical symptoms, right? Neurofield evidence for tau pathology emerges later. So currently, where Alzheimer's therapeutics is there are immunotherapies that target these amyloid plaques. And if you clear them away in people who are in the earlier stages of diseases, there seems to be some level of clinical benefit. That level, it's pretty modest. It comes with some substantial risk, and it's unclear whether that risk and the benefit are exactly worth it. But It definitely seems to do something. And so there's a movement to try to identify people with amyloid plaques earlier and earlier and earlier in the disease process so that you can get the greatest amount of benefit. That's secondary prevention or secondary treatment of Alzheimer's disease. So targeting the established pathology that underlies the neurocognitive impairment. If glymphatic function contributes to the clearance of amyloid and tau, and its impairment contributes to the development of amyloid plaques and tangles, then glymphatic impairment exists upstream and to the left of amyloid and tau on this Jack curve. And if that's true, then if you could identify people who have the glymphatic impairment, whether that's with an imaging approach, a fluid biomarker, or a device, you could identify people who don't have amyloid plaques now, but as you would with a fluid biomarker or a PET scan, but rather people who are at risk for the development of that pathology before it develops in the first place, right? And if you could identify therapeutic interventions that restore glymphatic function, you actually have the opportunity to prevent the development of those pathological features. So preventing the development of amyloid plaques, preventing the development and propagation of tau pathology. So this is getting at primary prevention, right? Sort of on the heart disease model. This is identifying people not who've had a heart attack, not who have atherosclerosis right now, but people at risk of developing those things, right? So those are people with high blood pressure or hyperlipidemia, and treating the risk rather than the pathology itself, right? So it may provide an opportunity for primary prevention of Alzheimer's disease and other neurodegenerative conditions, which is one of the reasons why people are so interested in this idea. So what about TBI? So in the last 5 minutes or so, we'll talk about a little bit of work that's been done in the traumatic brain injury setting. So we published a paper back in 2014 that showed that in a rodent model of moderate to severe TBI, there's impairment of glymphatic function and an impairment of tau clearance. And that if you impair glymphatic function, you exacerbate pathological features of tau in a rodent model of TBI. That's long published data, so I'm not going to really talk about it. I'm going to talk instead about some newer stuff. So we've been focusing on blast-related traumatic brain injury. So with the wars in Iraq and Afghanistan, and subsequently, the exposure to blast overpressure, both in military and even in civilian populations, has emerged as a substantial risk in U.S. military populations. Literally, tens of thousands, perhaps hundreds of thousands of veterans were exposed to blast during a deployment to Iraq and Afghanistan. The long-term consequences of that blast exposure remain unclear, partly because the people who experienced that blast, that was only 10, 15, 20 years ago. So those people aren't very old yet. So we don't yet necessarily know what's going to happen. There is, though, an epidemiological literature suggesting a relationship between military TBI and the vulnerability to dementia later on in life. And a lot of this work has been done by Christine Yaffe, who's at UCSF and at the San Francisco Veterans Affairs Hospital. So we wanted to know what does blast TBI do to the glymphatic system, and what is the consequence of this? So this is a study. This is a study we just published in Brain just a little while ago. This was a human neuropathological case series we conducted in collaboration with Dan Pearl, who's at the Uniformed Services University in Washington, D.C. So Dr. Pearl has access to a case series of veterans who died later, having had an exposure to blast TBI earlier in life during their military service. When we looked at frontal cortical aquaporin 4 expression in blast-exposed individuals and in control individuals, so this is the frontal cortex, this is the pial surface up here. Down here, you can see the subcortical white matter boundary here. This is the gray matter, white matter boundary. We see these two different things. We see laminar changes in aquaporin 4 expression, where we generally see enhancement in superficial layers. We also see enhancement both within the white matter and at the gray matter, white matter boundary. right? And you can see that both this is at the crest of the gyrus here, and this is at the depth of the sulcus here. And then when we measure perivascular aquaporin 4 localization, we do see changes in localization with blast. But the bigger changes, the more pronounced changes, are these laminar changes in aquaporin 4 expression, where we see higher levels of aquaporin 4 at tissue boundaries, both boundaries with the CSF, like you see here in the sulcus, and at the gray matter, white matter boundary, like you see down here. At our facility, we're at the VA Puget Sound. So there's a research center that focuses on blast TBI. We have a rodent model of blast overpressure. So you take a mouse and you place it into a shock tube where it experiences an overpressure wave that is similar in its dynamics to what a military service member would experience when exposed to like an improvised explosive device. So in this model, we took mice and exposed them to sham treatment, repetitive blast TBI. So these animals underwent 3 different blast exposures over the course of 5 days. So 3 blasts with 2-day intervals. 7 days post-blast, we didn't necessarily see a change in aquaporin 4 expression or localization. But at 28 days, we saw this begin to emerge, where we saw this, if you look at the subcortical layer, just below the peel surface, you see this enhancement right at the boundary between the cortex and the subarachnoid CSF space. This is now, mice are lissencephalic, right? So they have smooth brains, whereas humans have a very folded brain structure with gyri and sulci. This interface between the cortex and the CSF space, this is the closest thing to a CSF interface at the sulcus or at the crest of the gyrus that a human has. And so where we do see these interfaces in mice, we do see this aquaporin 4 enhancement. So similar types of changes in aquaporin 4 expression in rodents following blast to what we see in veterans with a history of blast TBI. In these rodents, though, we have the ability to measure glymphatic function. So here we're measuring glymphatic function with dynamic imaging of an infrared tracer. This is evaluating CSF tracer distribution in different slices. We're looking 7 days post-blast and 28 days post-blast. We do see what seems to be a progressive decline in this process, in this glymphatic exchange, where at 28 days post-blast, we see a pretty pronounced impairment of glymphatic function. And this timing coincides with those sort of laminar changes in aquaporin 4 expression that we see in blast. So these data suggest that we see a certain type of laminar change in aquaporin 4 in veterans who died with prior blast history. We see similar changes in rodents exposed to experimental blast. We see that those changes are associated with impairment of glymphatic function. But these data don't tell us that there is glymphatic impairment in humans. So to try to get at that question, we evaluated some MRI data that we have for evidence of glymphatic impairment. And here I have to use a little bit of a caveat. This MRI, this approach that I'm talking about, this isn't measuring glymphatic function. This isn't measuring fluid exchange and solute exchange. This is simply looking at structural features of MRI that we think are consistent with perivascular impairment. So the appearance of MRI visible perivascular spaces, particularly in the subcortical white matter, so these little dots, these are little vessel-shaped CSF isointents with CSF features in subcortical white matter that have originally, so neuroradiologists have noted these for a long time. Originally, they were thought to be benign neuroradiological features, but more recently, these features, which are called MRI visible perivascular spaces or dilated perivascular spaces or enlarged perivascular spaces or Virchow-Robin spaces, these are all synonyms for the same thing. These have become known as features of cerebrovascular disease. So both cerebral amyloid angiopathy, cerebral small vessel disease, they're seen in aging and they're also seen in the setting of hypertension. But they're also associated with chronic sleep disruption and sleep apnea and even concussions. And so using MRI data from a cohort of Iraq and Afghanistan veterans that we follow here in Seattle, we evaluated the question of whether the emergence of these MRI visible perivascular spaces, which we think reflect impaired glymphatic function, whether those occur with TBI exposure, and whether they're associated with the persistence of post-concussive symptoms. So this is a study in 56 veterans with a history of TBI. They're pretty young, so they're 30 to 40 years of age, usually a few years downstream of their last TBI episode. This is a multivariate model where we looked at the relationship between aging, military TBI, and other features. A couple things to note. First, we see the strong association between aging and these MRI visible perivascular spaces. That's a well-known thing, so that's a little bit of a sanity check. We do see a positive association between number of blast TBIs in the military and perivascular space burden. And interestingly, we see a statistical interaction between participants who have poor sleep. So these are people who on PSQI, so the Pittsburgh Sleep Quality Inventory, exhibit clinically significant poor sleep. Those people have a steeper relationship between traumatic brain injury number and perivascular space burden, suggesting that there's some linkage between poor sleep that occurs after TBI and this MRI measure of glymphatic function. We see that if we evaluate the persistence of post-concussive symptoms, and these are so this is years after the final TBI, we see that there is an association between perivascular space burden and the persistence of post-concussive symptoms like balance issues, memory issues, and cognitive issues, post-traumatic headaches, and things like that. So there does seem to be a link between not just whether they've had a TBI, but whether the clinical features of that TBI that persist long-term, whether those persist. If glymphatic function contributes to the clearance of amyloid and tau, then you might suspect that blast TBI through impairing glymphatic function could also be changing the neuropathological features of Alzheimer's disease in this population. So this is data from a study we just published in the journal Neurology. So in part of that same neuroimaging cohort and a group of non-TBI exposed controls, we have CSF biomarker data. So this is both amyloid and tau data from these participants. And what we observe is that within this population, we see that in the normal control population, A beta levels tend to increase over time out to about the age of 65. And after the age of 65, A beta 42 levels begin to decline. We also observe that total tau and phospho-tau tend to increase over time. What we observe is in the participants who are exposed to blast TBI, we do see, particularly in ages over the age of 45, we see lower levels of CSF A beta 42, lower levels of CSF A beta 40. We even see lower levels of phospho-tau and lower levels of total tau, although those findings are a little bit less robust than the findings that we see with A beta 40 and 42. So this decline in A beta 40 and 42 in the CSF, this isn't necessarily an Alzheimer's signature. Declining A beta 42 is an Alzheimer's signature, but typically A beta 40 doesn't decline. What we're seeing is a global reduction in CSF A beta 42 and 40, and maybe even tau and total tau. So what we think that this might reflect is actually the generalized impaired clearance of solutes like A beta and like tau from the brain tissue out into the CSF where it's detected as a biomarker. Interestingly, the decline in CSF A beta 42 in this population is associated with different cognitive measures, with a number of different cognitive measures. So we think it's clinically significant. What we don't know is whether those changes are going to result in long-term vulnerability to the development of Alzheimer's-related extracellular plaques and neurofibrillary tangles. All we see is that there does seem to be a CSF biomarker difference, and it does seem to be connected to cognitive function in this population. So to conclude the TBI piece, in human participants, so these are neuropathological cases that were exposed to blast, we do see these laminar changes in aquaporin 4 expression that are characteristic of both people and rodents exposed to blast. In rodents that have similar types of changes, we do see evidence of impaired glymphatic function. In veterans exposed to repetitive blast TBI, we see the emergence of MRI visible perivascular spaces that are associated both with poor sleep and the persistence of post-concussive symptoms. We think those are a measure of glymphatic dysfunction, although it's not a functional measure, it's a structural measure. And we do see that CSF biomarker levels are AD biomarker levels in this are reduced in participants with a history of TBI, and some of those reductions are associated with poor cognitive performance in these populations. So we think that in veterans exposed to blast TBI, they may be exhibiting evidence of impaired glymphatic function that may, in the future, predispose them to development of conditions like Alzheimer's or Parkinson's or chronic traumatic encephalopathy. So it looks like we've got a little bit of time for some questions. I'm glad to take them. This is my lab and our collaborators. This is, of course, our lab funding. Any group like this, I always do a shameless, come do a postdoc in Seattle, because we're always looking for people who are interested in this biology. So this QR code will take you to our lab's website and our contact information, where you can read about some of the other studies that we're doing. And we'd be glad to talk with anyone interested in collaborating or anyone interested in working with us. So thanks.
Perfect. Thank you so much for a very, very insightful presentation. So I will stop the recording.